Bace Study Journal by Layla Errol. Topic 1, Optical Density of Bacteria. Misunderstanding. I get confused on what the purpose of optical density is and how the optical density values differ and what that means for the bacteria. Corrections. To correct this misunderstanding, I did more research on the optical density of bacteria. I researched it and I also watched videos to help me understand the topic. After that, I looked if there was anything on the UF quizzes related to the topic and researched those questions. What is optical density? Optical density is a measurement that estimates the concentration of bacteria in a liquid. It uses a spectrophotometer to detect how much light is scattered by the bacterial cells when a beam of light passes through the sample. A spectrophotometer reads the wavelength of 600 nanometers. The importance of optical density is it helps determine a specific cell number and helps scientists develop/monitor bacterial growth over time. Optical density readings also are used with serial dilution and plate count. A high optical density means high concentration of bacteria, and a low optical density means low concentration of bacteria. Topic 2, different types of DNA. I get confused with what different characteristics go with the different types of DNA. To correct this misunderstanding, I did research and took notes on each of the different DNAs, which are prokaryotic, plasmid, eukaryotic, and mitochondrial. I also made flashcards to help me memorize the specific characteristics of each type of DNA. What are the different types of DNA? The different types of DNA are prokaryotic DNA, plasmid DNA, eukaryotic DNA, and mitochondrial DNA. Prokaryotic DNA is circular, long DNA with thousands of genes. It has 4 nitrogenous bases, a double helix, and is in the nucleoid region. The plasmid DNA is a cytoplasmic circular DNA containing only a few amount of genes. It has 4 nitrogenous bases, a double helix, and is located in the cytoplasm. The eukaryotic DNA is linear, long DNA with thousands of genes. It is found within the nucleus. It has 4 nitrogenous bases, a double helix. The mitochondrial DNA is small circular chromosomes. It only has maternal inheritance. It has 4 nitrogenous bases and a double helix and is found in the mitochondria. Topic 3 is protein chromatography. I get confused on what the purpose of chromatography is and what determines if it's in its mobile or stationary phase and what determines how the molecules separate. To correct this misunderstanding, I researched what protein chromatography was and watched videos and took notes on protein chromatography. Protein chromatography is a technique that separates, identifies, and purifies proteins in a mixture. The stationary phase is a solid material, while the mobile phase is a liquid buffer solution that flows through the column. Based on the molecule's degree of affinity for the mobile phase and stationary phase, as the liquid passes through, molecules will be retained and others will pass through. The solution flows out of the bottom of the column and is collected in a series of tubes. Each are set volume and are analyzed for the presence of the protein of interest. Topic 4, immune response. I get confused with the different types of immune responses and what specific cells help with the immune response. To correct this misunderstanding, I researched what an immune response is and the different types. I also researched the different types of cells involved in the immune response and watched videos on how the immune response works. Immune response is the body's way of defending itself against harmful substances and microorganisms. The different types of immune responses include innate immunity, adaptive immunity, cell-mediated immunity, and passive immunity. The primary immune response is the initial antigen exposure, usually 1 to 2 weeks. It rises in antibiotic production directed against the antigen. It's dominated by the IgM and the IgG production. The secondary immune response is secondary antigen exposure, more pronounced and rapid. The IgM is produced in days, followed by large production of IgG. There are T cells, white blood cells, B cells, and dendritic cells involved in the immune response. Topic 5, monoclonal antibodies. I get confused with what monoclonal antibodies are used for and the steps taken to use monoclonal antibodies. To correct this misunderstanding, I researched what a monoclonal antibody is and what it is used for. I also reviewed the questions from the UF quizzes related to this topic. I took notes and reviewed the notes, making sure that I understand the concept. Monoclonal antibodies are laboratory-made proteins that bind to specific targets in the body. An example would be antigens on cancer cells. The steps to injecting a monoclonal antibody is injecting an antigen into an animal, extracting the B cells and fuse them with myeloma to create hybridomas. Use assays like ELISA to identify hybridomas. Isolate and clone the selected hybridoma cells. Culture the cloned cells to produce large quantities, and use chromatography to isolate the monoclonal antibody. The recombinant DNA technology has yielded new antibody production methods. Study skills. Testing with flashcards and quizzes updates and strengthens the memory. Space the studying over various weeks, don't cram the last minute, and mix up the flashcards, don't use the same ones every time. I will use these study skills by preparing weeks before the Bace and reviewing my notes, as well as continuing to take practice quizzes and using flashcards to settle the information in my brain.